Um, Thank you very much. Thanks for the introduction, and thanks for inviting me to talk in RegSys in ISMB 2024. My title is Integrative Modeling of Multiscale Single-Cell Spatial Epigenome. It's kind of a very generic title. What I will focus on is some of our recent work in single-cell 3D chromatin organization, because you can view as the chromatin is their spatial organization of the DNA. So I don't really need to have any introduction to give any background to this audience. We all know that in our body, we have different cell types. They have different gene regulatory programs, but essentially they share virtually the same genomes. How these genomes are modulating gene regulation in different cell types, it's still not fully characterized and fully understood. And largely, the number of cell types is we have a lot of advances in the past decade due to the technology advancement in single cell sequencing, especially single cell RNA. So if you look at the example on the right-hand side, this is a recent work from the Allen Brain Institute. This is the whole mouse brain. See, they're almost running out of colors. These are different colors are indicating different cell types. But what's the functions and cellular functions of these different cell types in different cellular conditions? A lot of people are trying to understand, and certainly the epigenomes are important factors modulating and regulating the gene programs. And one important layer of the epigenome is, of course, the high-order organization of the genome, how the genomes are spatially organized in three-dimensional space. And if you went to the first invited talk in the HitSeq session given by Sushmita Roy this morning, you probably have already had a pretty good background information about how these genomes are organized in multiple scales, all the way from in very low resolution or high level. All the chromosomes have their spatial territories, and we know that chromosomes are largely segregated into active and inactive compartments, and people call them A and B compartments. I will refer to that later on. And if you keep zooming in, you see that chromosomes are also organized in these domains, and the so-called topologically associated domains, where the chromosomes are interacting more frequently with the chromatins in the same domain, and much less frequently with chromatin and DNA in other surrounding domains. And then because of this, you see these boundaries that between the domains, and then at the highest level, chromatins are also forming these loops, and it's essential for transcriptional regulation. And all these different structures and features are evident in 3D genome mapping methods like Hi-C, as you can see at the bottom. And you can perform different kinds of computational analysis to review these different features at different scales. So if you have interest, I listed the reference at the top with Frank Albert at UCLA and Tom Stelly at the NIH. We recently wrote a review article to go over different kinds of computational methods for identifying different types of 3D genome features, both in omics data sets and also the integrative analysis by incorporating some imaging data. So if you have interest, please take a look. So you may wonder, how does the 3D genome look like? Because look at in single cell, because almost all these features and analysis so far are done at the bulk level, at the population level. How does these 3D genome look like in single cells, and how they vary across different cell types? So first, you can you ask, why do we care about single cell 3D genomes? I think I listed here some of the critical questions in my view, and certainly there are more that are related to single cell 3D genome analysis. First, if it's from a technical standpoint, how can you develop reliable computational approaches to identify different types of 3D genome features in different scales? Because there are a tremendous amount of computational challenges that I will briefly mention in a minute. And this is much more difficult than identifying these features in the bulk level, in the population Hi-C. And second, very importantly, to understand the connections between 3 genome features in different scales with genome function. And there are different kinds of genome functions, and certainly transcription and gene expression is the one that most people are interested in. What is the structure function connections in different biological contexts? And if you look at the single cell, so one of the added value, of course, is that with the single cell, you'll be able to see these features and their variations in individual cells and this so-called cell-to-cell variability, and how these variability leads to genome structure and function, in particular gene expression observations at the population level, and how does the cell type specific 3 genome features modulate cell type specific gene function, especially in the context of health and disease, like developmental programs and also different kinds of disease progressions. So first, the computational challenges. What I'm showing you here, the bulk level, just like single cell RNA versus bulk level RNA, you see these signals that are aggregated in bulk level. The contact map you observe from bulk high-C sequence is like a smoothie you're looking at. But ideally, you want to unpack this and then try to identify the ingredients of that, and not just the ingredients, but also the relative proportion of different ingredients that are making the smoothie. So, with the single cell 3D genome mapping approaches, the goal, of course, is to find out, for example, in a complex tissue, what are the different types of structures that are present in individual cells, and what is the relative proportion of these different structures and their variations in a cell population. But it is tremendously difficult compared to 1D types of single cell assays, like single cell RNA or single cell ataxia, because those are, again, one-dimensional along the genome. These are, you want to represent these 3 genome features at least in a 2-dimensional contact map, and then the dimensionality is higher, and then in terms of a level of sparseness and also noisy data is much more challenging than the 1D single cell assays. And also, for these type of analysis, you're not only just trying to identify, let's say, how to represent a cell based upon their 3 genome features, some kind of cell embeddings for your cells so that you can perform follow-up clustering and other downstream analysis. But because we wanted to identify the variations of these 3D genome features, you also wanted to identify, really recognize, detect these features in individual contact maps. And if your signal is very low, it's intrinsically difficult. So I listed here a few of these different types of single cell high-C technologies in the past few years, and almost all are based upon proximal ligation, just like the high-C that you see in the bulk level. And but regardless of the experimental approaches, they share a lot of these computational challenges that I listed here. And 2 of the most important computational tasks, and there's some early work by people in this room. For example, how do you use the single cell contact map to represent a cell to do this type of embedding so that you can separate cell types according to their 3 genome features, and how you can, again, I'm repeating myself, how based on these very sparse contact maps, how do you enhance the signals by imputing, the missing entries, and so that you will be able to identify more reliably the type of features that I showed you earlier, the tags or the loops and then the compartments in these individual contact maps. Now, here's our one of our solutions, and then this was developed, published a bit over 2 years ago. This framework, we call it, the name is Higashi. We use a hypergraph formulation to model a single cell high-C contact map, a collection of single cell high-C contact maps. So very briefly, because this is already published, so each hyperedge connects different kinds of nodes in this hypergraph. So first of all, the hypergraph represents the single cell high-C data sets. So there are 2 types of nodes in this hypergraph. In hypergraph, you have nodes, you have edges. The edges are connecting multiple nodes, and they're hyper edges. And the 2 types of nodes in this hypergraph is they're either bin nodes, genomic bin nodes, or the blue nodes are the cell nodes. And then there are only 1 types of hyper edges that we're modeling, we're using, which is these 3-way or triplets. You have these triangle that you see that each triplet hyper edge connecting two genomic bin node and one cell node. So each hyperedge here represents the following. These two genomic nodes bins are interacting with each other in a given cell. So in a way, you are representing an entry in a single cell contact map, right? So now the problem is being converted into the following prediction task. If you're given a hypergraph like that, you have the nodes and you have the edges, the vast majority of the hyperedges are not observed because they're missing, because of the sparsity of the assay, and then you're not able to observe that. So your goal, the machine learning goal, is to predict, given what's already there, you want to predict the missing triplets, the missing hyperedges. So given this hypergraph, can you predict if two genomic bin nodes are interacting with each other in a given cell, right? So you're given a hypergraph like this, you're trying to predict the missing hyperedges. So we use the very similar to self-supervised learning scheme. So you can learn the model by your observed triplets, right? You can mask some of them out, and then you try to predict them, and then you can use the model to predict the missing ones. So these framework will allow you to generate the blue nodes, the cell embeddings that you can use for clustering, and also of course you will be able to predict the missing entries. So you can do two types of analysis at the same time with the same framework. I'm showing you how it works. On the bottom left, these are single cell contact maps from an IPS cell line WTC11. At the top panel, these are single cell contact maps, very sparse. And after you have imputed the missing entries, you'll be able to observe, at least some of the structures, like these domains, are becoming more obvious. And on the right-hand side, I'm showing you an example where this is after the imputation of the contact maps in different single cells. Each row here is a single cell, and each column is genomic bin. So you can see that we can clean up the contact map, and then you can derive this so-called insulation score, which measures the intensity of the domain boundaries. And you can look at these, different types of cell-to-cell variations of the domain boundaries across different cells. In this case, they're close to 200 cells. You can see that some of these boundaries are, even though at the population level, the pseudo bulk level, they all look like they're boundaries, but they have very different cell-to-cell variation patterns. And if you don't do this kind of imputation or removing the noise, it's the entire map, the insulation score map at the bottom is going to be very noisy. That prohibits this kind of more reliable analysis. Now, we also apply this to tissues. This is a data set of single cell Hi-C data from Joe Ecker's group at the Salk Institute, profiling the human prefrontal cortex. And at the UMAP at the bottom, this is only from the embeddings from the single cell Hi-C part. And you can see that we can separate not just only the major subtypes, cell types in this brain region, but also some of the inhibitory neuron and accessory neuron subtypes. And then, interestingly, if you look at these specific cell types, and then you focus on their marker genes, and then you compare, you look at their specific 3 genome features in that particular cell types, and you saw that these marker genes surrounding regions, they also show cell type specific 3 genome features in terms of AB compartment scores and also insulation scores. And you can clearly see that they're distinct across different cell types. Now, let's step back a little bit and go back to the bulk Hi-C analysis, because this is something that I want to highlight. This is our recent work. It's called Single Cell Ghost, is to specifically look at types of spatial patterns of chromosome organization in single cells. And you probably have heard a lot of these talks, and then if you look at these Hi-C analysis papers, you will see that people, the field is really obsessed or interested in identifying compartments in A and B, and A compartments are mostly genes are expressed, and then for the B compartments genes are repressed, and then this can actually be derived pretty straightforwardly from a simple Hi-C contact map analysis. If you take the correlations of all the rows or columns in the high-C contact maps, and then you do a simple PCA, as I show on the bottom left here, you will be able to see that the eigenvector and then first the principal component that corresponds to these A and B compartments. So it's a very natural way to reflect the important factor that separate these patterns in a contact map. And after the first high-C, this was reported in the first high-C paper in 2009, and a few years later, from Erez Libum and Aiden's group, they sequenced a cell line called GM12878, which is a major cell line in the ENCODE project and the various other genomics projects. They sequenced this in very, very high coverage up to 5 billion reads. And then if you sequence the high-C in very high depth, and then you'll be able to actually able to identify interesting patterns, not just for the intrachromosomal contact maps, but also for interchromosomal contact maps. So this is what they did on the right-hand side. If you take the rows are the genomic beings from odd number of chromosomes, and then the columns are genomic beings from even number of chromosomes, all the other way around, you'll be able to simply cluster this interchromosomal contact map into a few major clusters, and then the authors call them subcompartments. And on the I don't know if you can see my mouse here, and in this cartoon shows that this type of subcompartment patterns that you can observe from a simple clustering approach from interchromosomal contact maps. And these different subcompartments, A1, A2, B1, B2, B3, and these 5 major subcompartments, B4 is a small subcompartment of chromosome 19, and these major subcompartments very nicely correspond to various types of epigenomic features, and also other types of genome structure and function properties like replication timing or the contact frequency to nuclear laminas and so forth. So in a way that these subcompartments are definitely structurally and functionally meaningful and interesting, this just being obscured by a very straightforward AB compartment analysis, and then you're not going to be able to identify these more refined subcompartment structures. If you can further subdivide them into, basically, if you can subdivide A and B into smaller clusters, you will be able to review more interesting spatial patterns from the high-C contact map. And a few years ago, I have a student, he looked at this and realized that this type of interchromosomal clustering is not feasible for all the high-C data out there, because if the coverage is too low, then the signals in the interchromosomal contact map is simply not enough for you to do this type of clustering reliably. So the idea is that can you enhance the signals for the interchromosomal contact maps? So this is a very simple autoencoder exercise that you can train a model using GM12878, and then you can apply the model to some of the low and modest coverage high-C interchromosomal contact maps to recognize these subcompartments. And of course, what we are interested in now is can we do this in single cell level, right? Can you recognize the subcompartment patterns in single cell? So 2 graduate students, and both of them have already graduated and moved on, Kyle and Roche. So they came together and figured out a solution. And this is this came online about 2 months ago. We call it single cell ghost. So we wanted to identify single cell 3 genome subcompartments, and this really builds upon our early work, Higashi, that I presented early on. So we use both the Higashi imputed contact map, right? I kind of went through that briefly for the how do you do the imputations, and also the embeddings of the cells in the embedding space, because we're going to use that for each cell. You still wanted to borrow information from the cells that are in the same embedding space. They're similar to each other. You can kind of borrow information from each other to enhance any calculation that you want to do. And the method is embarrassingly simple. So we view, again, you can view the entire contact map as a graph where each genomic bin is a node in this graph, and edges are indicating whether these 2 bins are somewhat similar to each other, and by the imputed contact map. So the cell embeddings define the K nearest neighbor, and then it's a way that you're not just looking at the cell that you're dealing with right now. You look at neighboring cells in the same embedding space so that you have more stuff to to handle, and then you can enhance your sampling data sets. That's what we did. And then for each genomic locus, you generate an embedding for a genomic bin. And I'm going to elaborate a little bit how we generate the embeddings for the genomic bin, because right now we have a very specific task. We wanted to look for genomic bins that are very similar to the one that you're looking at. But when you learn the embeddings, you also try to minimize the similarities between the bins that you know are very different from each other or very far away from each other spatially. So we devised a very, very straightforward and simple sampling approach to do that. You're not sampling, or you're not trying to go over all the possible paths in the graph, in the graph, but you only focus on the part that are, the bins are very similar to each other versus the bins that are very different from each other. And once you have generated those walks or random walks in the graph, that becomes just like the sentences just like a sentences, right? Each word now is a node, and you can use these very widely used approaches, the node to vec, for example, or word to vec, to generate the embeddings for the entities that you're looking at. So here's the high-level summary of the approach. So at the top, you can only look at the top, because the bottom basically is an elaboration of the same approach for the neighboring cells in the embedding space, right? So at the top, we do this you look at what we call the positive walks between the top quantiles according to this contact map, and you sample the walks from the nodes that are very similar to each other. And you also have these bottom quantile. These are the genomic bins that are presumably very, very different from the ones that are currently looking at. And you generate these random walks. So you can start from your current node, and you look around, right, to the neighbors, and you walk to the next one. And this is the first order, of course. And you can do this for the second order. You can look at the neighbor's neighbor. And you do this for many, many different random walks. And then those random walks generates a strings of nodes that you can deal with. So these strings, the walks, are coming from the walks of nodes that are very similar to each other. Also, the walks are generating the walks between other genomic bins that are very different from the ones that you're currently looking at. And once you have these walks, you can feed that into a neural net to learn the embeddings of the nodes. And there are some additional steps to ensure that between different chromosomes and different cells, these subcompartment annotations, and the subcompartment annotations are a straightforward clustering of the embeddings. But there are some steps to ensure that these subcompartment annotations between different chromosomes and different cells are comparable to each other, so that in the end, in different cells, in different chromosomes, you'll be able to generate a generic cluster IDs for different kind of regions. So I'm showing you some analysis results to show you, they're reliable by, nothing is direct. They're all kind of indirect evidence from other types of genomic data that we can use to support the identifications of subcompartments in single cells. The first one is on the left-hand side. This is in GM12878. I'm showing you a type of evaluation that we did to show you that the subcompartment annotations, if you pseudo-bulk level aggregated from single cell, we observe that they are actually more reliable than the population level annotations. So this particular row here, this is the bulk Hi-C annotations from the original publications. And this is the contact map, both the pseudo-bulk level and the original bulk level. And at the top, these are single cell subcompartment annotations, aggregated ones. And you will see that, in this particular column that we highlighted, in the bulk subcompartment annotations, there's nothing there, or they're all B3 subcompartment. But actually, if you look at the detailed contact maps, both at the pseudo-bulk level and in the bulk level, you see that there are some differences here. So it's much more sensitive than bulk level approaches in identifying these subcompartments the take home is that if you do the subcompartment annotation in single cells and aggregate them, it's actually going to be more reliable than the ones that are directly applied to a bulk level Hi-C. And I think now the evaluation on the right-hand side is a bit more interesting. So we show that not only this approach works for single cell Hi-C data, it also works for imaging data. So this is a type of imaging data where it's a DNA merfish. You basically image the chromosomes, and you can turn this image data also into a contact map, right? And by the way, this is from an earlier publication from Xiaowei Zhuang's group. So this is in MR90, and this chromosome imaging, so you can view each image as a single cell contact map. You do the same types of imputation and so forth, and you predict identify the subcompartments. And once we have the subcompartment for individual cells, you aggregate them into pseudo bulk level subcompartments. And you can compare with bulk level epigenomic features. And the heat map here I'm showing you, this is actually very consistent from bulk level subcompartment analysis by comparing subcompartment annotations with various types of epigenetic features like histone modifications I'm showing you here. The active marks are mostly in A1 and A2, and some of the more repressive marks like these constitutive heterochromatin marks, H3K9 trimethyl, is mostly in single cell in B3 subcompartments, but there is also this K27 trimethyl. This is the facultative heterochromatin, is mostly in the middle, like B1 and a little bit in A2. And if you look at the replication timing, it nicely separates the replication timing, in S phase from early to late, according to their different spatial positioning in the nucleus. And then we apply this to tissues, and this is same data set I showed you earlier. If you only use the single cell AB compartment scores, and you see that a lot of these subtypes are still, you cannot separate them that well. But if you use the single cell ghost embeddings, you can separate them better according to their subcompartment features. And if you focus on the marker genes for these different cell types, and you see that in the marker genes, they're typically in the active subcompartments, and then in the rest of the cells in the second row in each cell type I'm showing on the right-hand side, they're much more inactive. So recently, there is this DIPSY technique that allowed for haplotype resolved chromatin contact maps via genotype phasing and also some imputations. And this gives us the opportunity to look at different kinds of 3D genome features at a allele-specific resolution. And so we just took the allele-specific 3D genome contact maps and then derived subcompartments there. And on the left-hand side, this is a similarity distribution of subcompartment annotations between the two alleles across developmental brain different stages. And so as we saw, there's a decreasing similarity scores during the developmental stages. And on the right-hand side, we also found that the regions around the imprinted genes are much more likely to have allele-specific subcompartments. So all these, on this slide, very, very briefly, this basically shows us that this type of analysis, that the subcompartment annotations in single-cell 3D contact maps, and these type of analysis really demonstrate the connections between subcompartments and different layers of genome functions. But if you realize that so far what I presented, this type of analysis are between structure and function, they're almost always indirect, like at the bulk or cell type or pseudo bulk level, and it will be ideal if you have these two modalities, like the chromatin contact maps and gene expression coming from the same cell, so that you can directly compare them and then study their variations and correlations in different in different contexts, and that will be essential. So this is what we did recently. So in the past few years, we had a close collaboration with Zhijing Duan's lab at University of Washington, and so developed his lab developed the experimental assay of what we call GateSeq, which is a co-assay of single-cell Hi-C and single-cell RNA. And this work is led by a graduate student in my group, Tian Ming. So he did all the troubleshooting in the development stage, and also all the integrative analysis that we did later on. I won't go through the technology itself. It's recently published in a few weeks, I think in late May. If you have interest, take a look. I'll just quickly go through some of the highlights of the analysis, the integrative analysis, especially by integrating this type of data with other types of single cell assays and the new insights that you can obtain from those type of integrative analysis. So first off, just show you the quality of the data. Both the single cell RNA part and the single cell Hi-C part are having very high quality. This is in a mouse brain, a bit over 3,000 cells, and each cell we have a few 3,000 genes, 2 to 3,000 genes, I believe. And for each cell, we all actually have over 200,000 contacts. So it's not particularly high coverage, but it's already pretty high coverage, and 20,000 UMIs for the RNA part. So you can use these two modalities to assess how they perform, right, in distinguishing different types of cells in this mouse brain region. And we found that both RNA and then single cell Hi-C are able to distinguish very fine scale cell types in the mouse brain, and 15 accessory neuron subtypes, 8 inhibitory neuron, 8 and 5 glial, and overall there are 28 different cell types that can be identified by both RNA and single cell Hi-C part separately. And of course, you can, once you have done that, you'll be able to, in the subtype level, you'll be able to directly connect these two different modalities. And even within the same same cell type, you will be able to see this structure function connections and their variations in the same cell type. And this slide shows you this connections, the multiscale connections between 3 genome features in different scales, loops and compartments and domains, and their gene expression. And on the left-hand side, these two plots showing you, in general, the connection, because these two modalities are from the same cell, you'll be able to generate this kind of plot, right? So you can look at the connections between gene expression and then the large-scale AB compartment patterns. And in general, we observe that there's a positive connection between AB compartment scores and gene expression. So if the chromatins are in more active domains, and then the genes that are on the chromatins are expressed higher. In general, that is the trend. And in terms of the installation scores, the domain, the intensity of the domain boundaries, it seems to be that in mouse brain there's an anti-correlation. So the installation score is lower, and then expression is higher. And then we highlighted one gene, this ERBB4 gene. This is actually a cell type specific gene in a cell type called PVALB, P-V-A-L-B. I highlighted on the slide. This is an inhibitor neuron cell type. And so this just gives you an overview of the comparisons between inhibitor neuron and accessory neuron, and also between PVALB and other inhibitor neuron subtypes. So the first column here, this is the single cell AB compartment scores. You see that in inhibitor neuron, this part this score is positive, is A compartment, but in accessory neuron, it's B compartment. And this is consistent with the understanding that in this region it's supposed to be more active. This is probably not particularly inspiring. And if you zoom in, you see more refined correlations between installation scores. This is in PVALB, the installation score is lower than other inhibitor neuron subtypes, and this is also being reflected in the contact maps. If you keep zooming in, we actually identify one chromatin loop in PVALB, but it's not present. The loop is present in PVALB cell type, but not in other inhibitor neuron subtypes. And then we integrate with some of the other single cell multi-omic data. In this case, this is I believe it's a paired seq from Bing-Ren's group, so that they annotated these. It's a co-assay between RNA and chromatin accessibility, and they annotated an active enhancer surrounding this region. So we also have some additional analysis in the paper where we look at, if you integrate this type of multi-ohm co-assay between RNA and attack, together with this multi-ohm co-assay between RNA and single cell Hi-C, you'll be able to enhance your ability to connect distal regulatory elements with target genes. So it's kind of makes sense, right? Because if you only have the multi-ohm coassay between RNA and TAC, you don't have the high C will give you the chromatin interaction, the loop information that will enhance your ability to connect the regulatory element to target genes. So this is very briefly. So we in the mouse brain we have this gauge-seq data, but there in the same region there was some spatial transcriptome data in Murfish published a couple of years ago, and so we had this thought, whether we can integrate them. You can use the RNA part as the bridge. Essentially, you can overlay your single cell high C on top of a spatial tissue context using the RNA as the bridge, and to show you that this is possible using the RNA part as the bridge. As I show you we focus on this layer, this L5, and as expected, these L5 marker genes are expressed highly in this layer of the brain. And then you can see that the single cell AB compartment score also high in this layer. And then we did this for some of the other features, not just the single cell AB compartment score, but the insulation scores and gene body interaction scores. You can see that most of this is correlated as we expected, but there's some differences in certain regions. For example, in here, it looks like the AB compartment score is still relatively high, but the expression of the marker genes are low. This shows you the spatial variation of 3 genome feature that can be observed through this kind of integrative analysis. How about temporal changes? So to do that, we also generate the gauge-seq in hematopoietic differentiation. Showing you here is the lineage we focused on for 4 cell types. It's about over 800 cells, and you can see their trajectories. And by looking at these over 800 cells, you can do the following analysis. You can, according to their features over time, and for both RNA and single cell high C, you can cluster the genes according to different combinations of these temporal patterns. And we found that there are around 10 different patterns, and some of them really surprises us because we thought that these features, expression and AB compartment score and insulation score, they're going to be pretty consistent, right? But it turns out that there are certainly groups of genes that show very discordant patterns. For example, the gene expressions are going up as during this trajectory, but like AB compartment is going down, which is contrary to the observations in many other contexts. The reasons underlying this we don't quite know, and then that's something that we'll be looking into. So just to summarize before I wrap up. So in my group, we also have a lot of interest, not just in the spatial organization of the chromatins, but also the spatial organization of the cells and complex tissues. And then last year, we developed this method called spice mix, and then and actually made to the cover. And this is a spice mix, if you don't realize. And then the reason that we name it spice mix is the following. And the first author, co-first author, Ben Tedesda, is actually in the audience, and he came up with this idea. Just like spice mix, if you put different proportions of spice together, you get different flavors, and the cells may function in a similar way. There are some underlying factors, and if you put different proportions of these underlying factors together, you give rise to different cell states. Of course, the challenge there is that we don't know these different factors a priori. You have to learn them from your data. And then if you went to Sushmita Roy's talk in HitSeq this morning, she talked about non-negative matrix factorization. We actually use a very similar framework of NMF, but we integrated into a graphical model so that you can explicitly model the cell-to-cell connectivity in the tissues. And one of the advantage of this approach is, again, is like in Sushmita's talk, she mentioned that, stressed that the interpretability of these models, that you can directly understand, the contributions of these different factors towards different types of cell identities that you're identifying. And here we are looking at these, we call them metagenes, right? So this alpha metagenes in Liger, these are spatially resolved or spatially variable metagenes. And some of these metagenes you realize they like to hang out with each other in the spatial context. And some of them are more, what we call more repellent with each other. You can certainly do this for different cell types as well. And then right now we have a more advanced version of this, this spikes that have a next generation spikes that you can actually model temporal spatial transcriptomes, or at least the two samples, right, between treatment and control, or you have a temporal changes over time. You will be able to identify these spatially variable metagenes over time. So just a summary. So we're looking at these chromatin organization, how these DNAs are spatially organized with each other and to regulate gene expression. But the cell nucleus is only one part of the story. There are other components in the cell. And of course, the cells are also spatially organized in complex tissues. So how do you model all these different entities at different scales all at once, right? And then, we certainly need new technologies, but I think we also need more integrative computational approaches to really harness the power of these different types of information at different scales. And I really like this title of a Oscar-winning movie last year, Everything, Everywhere, All at Once. And this is the foundation model that we are talking about, right? You try to cover everything, and then the model itself can do a lot of interesting things. And so I think I'm running out of time. And so I'd like to thank the people who did the work. I think I highlighted their names when I introduced the work, but none of this would be possible without the collaborations with many. We are involved in several consortiums, and the work that I talk about today is most relevant to the NIH-40 Nucleum Consortium. And thank you very much for your attention.
Thank you, Jian. We can get one quick question, maybe. We are a little bit over time.
So just thanks for the very exciting talk. So I just have one quick question. So I really get excited about the deep sequence. Oh yeah. So the gauge sequence, I really hope that can be available for all the studies, all the data set. But in reality, that's not the case, right? Often the case you only have single high C and single RNA-seq, right? And that becomes essential for doing maybe multi-omics integration. There's a lot of existing tools available, but often the case, the multi-omics integration is done between RNA-seq and TAC-seq. I haven't seen a lot for RNA-seq and high C, right? So, you can align the probability distribution, but at the end of the day, you still need an anchor to align the high C cell, representation and RNA-seq single cell representation. So maybe, I guess the naive idea is you can assume maybe more interacting is associated with more highly expression. But
yes.
I'm not sure.
Some people use the gene-body interaction as a way to, there is an observation, at least in certain tissue context.
Have you tried that in your
We
studies?
We didn't try that, but I know, I know, kind of, right? We look at the gene-body interactions as a way, especially in the integration. But our experience is that it may not apply to all the contexts. In the brain region and in mouse brain, it seems to be quite obvious that there's gene expression is high. There's more gene body interaction from single cell high C. I don't know if this how generic that is. Actually, I know some people in there. Yeah. You want to say something?
Do you think is it risky to assume that can be used as loss function to optimize, say, the alignment?
I don't know, but I think the more co-assay data, between these different modalities will give the opportunity to develop those, what do you call it, multi-model, even they're not co-assay, like a separate single cell, and try to integrate them, right? This is an active area of research for single cell attack and single cell RNA. And I think there's an opportunity for other types of modality, even, multiple modalities integration, like you were alluding to. Perhaps it's more cost-effective or easy to do and more accessible for people to generate single cell for different different modalities and then integrate them rather than producing this type of, very sophisticated multi-model single cell assay. I don't know if that answers your question.
Yeah, yeah. All right, thank you.
Thank you very much.